What's going on, guys? I'm Shahab and today I'm here to explain for you about genes and proteins. Every processes from genes to proteins. What will happen in these parts? So at first we have some topics, a structure of nucleic acids and mRNAs and tRNAs, all parts, all types of RNAs that involves in translation and transcription. So, two important parts of genes to proteins are transcription and translation. So, at first, the role of RNA in translation. What is its role? Why the cell will use RNAs and different things and not using DNA? So, we have different types of RNA, which mRNA. mRNA is a messenger RNA which acts as a message from nucleus to cytoplasm of cells, and also it send the message means informations from nucleus to ribosomes. And also we have tRNA, which is involved in translation. It translates codons to proteins. And also we have rRNA, which it involves in ribosome. So now about the genetic code. We have codons. We have 20 types of codons and actually standard amino acids, 20 standard amino acids, which these amino acids would produce different proteins to form a molecule in cell. And also these genetic codes, which calls codons, there are 64 in cells. And now about how to read a triplet codon. Or triplet codes. Actually, the tRNA will read it, and it's transferred. The message informations will transfer by mRNAs to ribosome. However, only one reading frame is used to translation. Means only we have one frame. And now about the structure of tRNAs. tRNAs are an important part of translation in actually protein genes to proteins. So tRNAs are actually 70 to 80 nucleotides, made up of 70 to 80 nucleotides, and they have clover leaf parts. An structure like this that is called clover leaf secondary structure. And actually it's look like and yeah. Now about ribosome composition. We have different types of ribosomes in and actually eukaryotes and prokaryotes. They have different things, different parts. So actually first ribosomes are RNA protein supermolecular complexes. And they are most abundant type of RNA protein. There are some proteins, some molecules that are made up of RNA proteins. And their composition in prokaryotic and eukaryotics are different, and actually is composed of rRNAs, and actually rRNAs comprises 60% of ribosomal mass. So now let you go to learn about mechanism of DNA replication. How do DNA replicates? So at first to have DNA or genetic information in all generations, we should to replicate them. Actually cells should to replicate them. It is called semi-conservative mechanism. Why does it called semi-conservative mechanism? Because from its actual separation, one of the strands, actual DNA is double helix stranded, and one of those strands are all conserved, and one of that is half conserved. And actually, yeah, let me go for about DNA synthesis at the replication fork. There would be a fork when do DNA or DNA strands want to separate.
Pace that the replication is taking place is called the replication bubble. And in circular DNA, like the bacterial DNA, we have only one origin of replication. But in linear DNA, like in eukaryotic cells, like us humans, we have several origins of replication because we have a very, very long DNA. And as you see in this figure, we have two strands that are made. We have a template strand at first, which is the DNA that we have. But the DNA that is made newly is made up of lagging strands and leading strands. And the leading strand is actually the long part. We know that the DNA replication takes place from the 3' end to the 5' end. So we have the replication from the middle of the bubble. So what about the other side that is empty? We will have some lagging strands that follow up, and these will have the overlapping fragments that I will explain more further. So let's take a molecular view at DNA replication, as you see in the figure. We have an enzyme called helicase that unbinds the DNA. So we have the DNA strands unbinded from each other. And then there are some molecules, some proteins that hold the DNA from binding together again. And these are called the single-strand binding proteins. They stabilize the DNA strands because the DNA strands want to bind so badly. And step number 3 is that we have some DNA polymerases that form primers. DNA polymerase 1. I'm sorry. I made a mistake. Oh, I'm going to cut this part. Cut this part, please. Thank you. Okay, as I was saying, some enzyme makes some primers on the template strands, and these primers are the reason that DNA polymerase 3 could make the rest of the DNA. So, as you can see in the figure, this is the DNA polymerase 3 that is making the leading strand. And so, if we take a closer look, we can see that it should start from the 5' end. So, it should start at the 3' end. So, we need a primer that has the 3' end. Okay. And let's take a look at some enzymes and their functions. Let's take a review. We said that helicase unwinds the DNA from its double helix. And we have some proteins called single-strand binding proteins. These stabilize the single-stranded DNA and made them that so they wouldn't bind together. And we have the primates. This is the enzyme that actually makes the RNA primers from the 5' to 3' end, so that DNA polymerase 3 can start making the DNA strand. But if you pay attention to this process, the DNA replication, you will see that there are some empty spaces between the kind of leading strands or the lagging strands. And there is one enzyme called DNA polymerase 1 that attaches these empty spaces between the primers and the newly made strands. So we have another thing called DNA ligase that joins DNA strands too. But let me tell you something. What if we reach the end of the DNA, so we won't have any 3' end for the DNA polymerase 3 to make more DNA. So what's going to happen to the end of the DNA? The answer is nothing. The DNA is going to be uneven, and it's going to become shorter and shorter after every replication. So what will happen to this DNA if it becomes shorter and shorter in every replication? What happens to the embryo if this happens? The answer is that we have a very long sequence called the telomere. This is kind of a 6 nucleotide sequence that repeats for about 100 to 1,000 times. It differs in different organisms. So, every time that our DNA replicates, this telomere becomes shorter and shorter. But what if the telomere ends? And this is why scientists think that when this telomere ends, this is why we get older or maybe we die. But will it hurt the embryo if the telomere becomes shorter and shorter after every replication? Wouldn't it be so soon to end? So the answer to that is that there is some enzyme called telomerase, and that enzyme is repairing the telomere in the embryo state, or maybe in some cells in grown humans or grown organisms, but not in somatic cells. Should I continue? Okay, now let's talk about chromosome structures. If I want to say in abbreviation.
So, ladies and gentlemen, I'm taking over here to actually talk about the continue of mishandling she explained I think so until end of DNA replication. So now we have our DNA, which will actually continue to our messenger RNA. Our messenger RNA is for actually strands which can encode proteins, and the other ones which can't encode are in the non-coding RNAs. Yeah. And now we have the translation step, the process in which our ribosome into our cytoplasm, or endoplasmic reticulum actually synthesize our proteins after the process of our synthesis of RNA from DNA. And now, they will synthesize proteins from these, and all of this process is called as gene expression. This translation step is divided into 3 steps by itself, means initiation, elongation, and
now I want to continue with Mr. Kushek, which about the second step at the genes to protein means translation. So that the second step in getting from the gene to a protein, well, it will take place in the cytoplasm. Okay, it means it will go out of the nucleus. Now, here you know that the mRNA contains that information to build actually to make a protein. Yeah, the mRNA actually here we have a machine that's called as ribosome. So the ribosome is the researcher that is mRNA and some proteins. Well, the function of that is reads the sequence.
Hello guys, this is the continue of this discussion is about the translation and transcription, and in the end we have the post-translation and modification. Actually, to produce the protein we need these steps. It means transcription and translation. The transcription means the process in which the RNA is made from the DNA. And this is actually made by the RNA polymerase. RNA polymerase go on DNA and makes the RNA. And after that, in translation, RNA will go out of the nucleus, means in the cytoplasm, and within the ribosomes will make the protein. But how? Actually, within the ribosome, there are 3 places where another type of the RNA, which is called as the tRNA, is placed. And this type of the tRNA, it has an amino acid in its head. Actually, every type of the tRNA has a different amino acid. The different amino acids are 20 amino acids. So we have 20 types of this tRNAs that they will attach to our ribosome. But how this actually polypeptide of the protein is made? Actually, we say that we have 3 places of these tRNAs in actually our ribosome. And the DNA and RNA is going to this ribosome and placed in somewhere. And the special types of the amino acids based on the codon, which is the 3 bases on the RNA, will come into our ribosome. And also, these signals that are between the RNA and the amino acids that are coming, it makes the polypeptide change because every size of these places, which are 3 places in the ribosome, they will attach each of their amino acids. For example, thymine or another types of amino acids that will attach to each other. So first, this is the actually first structure for a protein. The protein is actually lots of the polypeptides together. We have dipeptide, which are 2 amino acids, and also tripeptide, which are 3 amino acids. But when there are more than 2 amino acids, we call it as a polypeptide chain. So the first structure for an amino acid, which is it has a carboxyl group, it has an amino group, and also a hydrogen atom, a central carbon atom, and R group, which is different in every type of the amino acid. And then in the secondary structure, actually it has 2 types of the structures, which is as cylindrical or as a sheet layer. Each of them are the secondary structures that are different in every type of the polypeptide, based on the type of the polypeptide that we can make. So the protein is made from our ribosome. And after that, we have the tertiary structure, which we have different domains of our protein in those chains. And so we can understand in here that our proteins can't be used after our translation immediately, because they must have a process called as a post-translational modification, which is after the translation and protein production. And it can be when to another type of regions of the body, for example, for the neurotransmitters, or for the cell membrane, or for every part out of the cell.